Okay, I'd like to welcome everyone out for our first annual pitch event with our CTOs. I will introduce Mike and Kevin here in a second, but just to kind of let you all know the purpose of this meeting, you all are doing great research, and part of the EPSCoR project is to build capacity. And one way to build capacity is to think about how your research can go beyond what you do in the academic lab. And Kevin and Mike are well experienced in helping navigate that trajectory. In the role of CTO, they work with other academic offshoots, different IP pieces, different students, different faculty members who have gone down that commercialization journey. This is also an opportunity for you all to get a glimpse of what each other are working on. And Mike and Kevin might have some questions for your work, some suggestions of how to push it along in a direction that it could be applied to industry. And it's also an opportunity for them to get just a perspective of maybe what labs to focus their efforts on and who to coordinate with. So I will let's have Mike, you go first, just introduce yourself, and then I'll have Kevin do the same.
All right, thanks. Pleasure to be here. My name is Mike Roth. I'm an ISU graduate. Did my PhD with Jim Roth out at Vet Med. Came out of Vet Med, and some of my research got purchased by a small startup company called Noble, and I was with Noble for 3 or 4 years. And then Noble got acquired by a German company called Boehringer Ingelheim. I was with BI for 25 years, had different roles from scientist, project leader, group leader. The last 10 years there, I was the global head of R&D, coordinating research efforts between Shanghai, Germany, and the US. And then 3 years ago, as Nigel said, had the opportunity to come back to campus to be a CTO and work with faculty who are doing research on vaccines, diagnostics, and immunotherapeutics.
Thank you, Mike. Kevin, why don't you give an intro?
Sure. Kevin Moore, just like Mike, I'm really happy to be here and meet this group. Chemical engineer by degree, just a BS degree. I was 30 plus years in industry, the majority of that with Archer Daniels Midland Company, where I did stints in R&D operations and also ran a couple of businesses. Just signed up for my 3rd year in this role as CTO, and it's been quite a blast. I should say I had no prior association with the great land-grant university that is Iowa State, and it's been a lot of fun just getting around campus, getting to know faculty members and what they do, and look forward to hearing what this group does as well.
Excellent. Thanks, Kevin and Mike. Appreciate your time being here. So I have the list of everyone who submitted their slides. We have 13 of them to go through. Just kind of ground rules. We'll try and keep it to 3/4 minutes. Just brief overview of what you're doing. We'll then have some time for Mike and Kevin to ask questions. And I just have them here in order as what was submitted. So I guess that's strategic next time. If you want to go last, you can have a file name that starts with Z, or maybe I'll mix it up and you won't know. Okay, so first off, I got Ali Riza. And again, I'm just gonna kind of pop them up, and you can unmute yourself, show if you have video capability, please, so that way we can see you and get to know you. All right, let me get Ali Riza's here.
Hello, everyone. Do you hear me, Dr. Nigel?
Yeah, you sound good. Let me just make sure I got your slide
Yeah.
available here. It's loading. All right. Share. Okay, and I'll make it big so everyone can see it. All right, take it away.
Yeah, my name is Ali Reza, and I'm working in the Dr. Cochran's group. My research project is about improving or toughening mechanical property of the PLA-PHA composites by a suitable compatibilizer. Well, PLA and PHA both are biocomposites, and we want to mix it and blend it together to replace the petroleum-based composites. Right now, as we know that one of our challenges in today's world is the plastics that has increased tremendously, and by 2050 we have more than 33 billion tons of plastic waste, and I think based on the reports, only 9% of them are recycled. So one of our chance and opportunity to replace them is using bio-based composites and plastics to replace them, and we can they are degradable and biocompostable. So what we can do is using these two promising candidates as the biopolymer, and use it through the extruder and make it good composites. And there are several applications for that. One of them is the packaging industry and consumer products and all other construction and automotive industries. Right now, I have some results, as you can see here in the two pictures. They are CM pictures of PLA PHA blends. The way it consists 8% of the PLA and 28% of PHA. As you can see, there is completely different, texture in the picture, and the rough texture is from the PHA, as is a rubbery polymer, and the smooth one is for PLA. It's a brittle. Somehow we want to replace the polyethylene or other famous petroleum-based composites with these ones. So we want to add and increase the mechanical, thermal, and also morphology of the polymer. So this is 3 minutes of description of my research project.
Great. Mike or Kevin, any questions or suggestions?
Yeah, so good project. Outside of the SEM, what do you anticipate the benefit of the composite over the neat PLA polymer? PLA is pretty widely available right now, PHA not so much. So what do you anticipate performance-wise the benefit of the composite?
Yeah, PLA is pretty brittle. We use PHA as a rubbery to add some, rubbery compound through the PLA and to increase the, the mechanical and thermal property and reduce the brittleness. So as you see in the CM, it's because we did not use any compatibilizer or additive to these composites. We don't have any, complete mixture of it. It's completely separate from each other. You can see it from the texture, but as we can add some suitable compatibilizer like dicumul peroxide or what else, I'm working on that. We go through it and makes it a complete rough texture which has a good thermomechanical property.
Great. Mike, if you have a question, I'll start queuing up the next slide.
Go ahead.
Nope. Okay. Okay, thanks, Alireza.
You're welcome.
Sorry, I was muting myself. Sorry, next one's from University of Iowa. Nick?
Yeah, I am Nick Brzkowski. I think mine's going to be just a little bit different than probably most of the other research here. This falls under the social science umbrella, but like we're more interested in getting people in biomanufacturing careers. So I'm part of the SCCT group, and I work directly with Nick Bowman from the University of Iowa, but Mary Loesch, Kyle Andres, and Hillary Wahey are all from the University of Northern Iowa, and they are also on our team. So we have a couple of main focus areas, and first of all is to understand the biomanufacturing industry as a whole. This is kind of what we've noticed so far is that dissemination of this information, like down into K-12 schools, into students and parents, that language isn't maybe conducive to everyone there, and so getting students into those careers hasn't really been happening. But what we want to do is determine persistence in STEM then as well. And all of this is happening through this social cognitive career theory lens, which has roots that was developed by Alex Bandura back a long time ago, but and that uses self-efficacy, outcome, expectations, and then personal goals to do that. Where can our findings be applied? We want to hopefully help increase awareness of, interest in, and persistence in STEM directly with high school and college level students, so that we can increase, diversity and personnel in the biomanufacturing industry, but then indirectly with faculty, parents, and guardians, and within the industry, especially with that language piece. Data collection, we've actually just started. So we're doing in-depth interviews now with industry professionals and community college instructors. Later this spring, we'll be sending out assessments to college and university faculty. And then in the fall, we'll start our assessments with college students, and then also high school students and parents. We'll do some surveys and then some focus groups with them.
Great, thanks, Nick.
You're welcome.
Feedback, Kevin or Mike?
Nick, this Mike, this topic is extremely important from the industry side, where they're looking to hire these people. What
Right.
industry professionals or companies have you connected with so far?
That's a really, I don't have access with that, because with the UNI, Mary, Kyle, and Hillary, they're the people that right now are doing the interviews. We are in those of us at the University of Iowa, we're kind of in a little bit of an IRB issue. I will just leave it at that. So I can't touch the data yet.
Okay.
But we're a little centered to I think they were with some section like within animal science biomanufacturing, I think. I don't know that I'm saying that correctly. I'm sorry, but it seemed pretty targeted.
Yep. If I'll just make an offer, if you or somebody on this team would like to be connected with biomanufacturing experts, I have a network of about 80 different companies, and would be happy to make those connections if your database needs it.
Okay, I'll keep that in mind. Thank you.
I'll just chime in here, Nick. Know that workforce development is part of the scope of what Mike and I get involved in. Mike a little
Okay.
bit more right now than me, but I'm just a little behind Mike. I heard it was either Mary or Monica Lamb from Iowa State just talk about the description, the level of understanding among parents, right? What biomanufacturing is.
Right.
Parents know STEM. Parents know there are jobs in STEM.
Right.
Parents know computer science, or they know healthcare or medicine. Biomanufacturing is not quite so tangible, and I'm
Right.
wondering if you've formed an opinion already, or have ideas around the marketing. How are we going to make this more tangible?
Have I personally formed an opinion, or has our team? Because that's
Yeah.
two different questions.
All right. Yep.
I was before my role, I was a teacher, and I think what's best for our students is to for them to understand the why. Why is this something that I want to do? How does this relate to my values? How does this relate to my outcomes as a professional? So for them, they don't want they want to see something that's more user friendly per se. They don't want a 2-page handout of all the things that that we can give them in all professions. So I don't know. I think it's more of those outcomes of what students want to experience after they graduate, besides more of like, what are your interests before they get there?
Great, thanks, Nick. We'll move on to the next.
Yep.
Yeah, just you were worried it didn't have application, but I just left this Iowa Bio Conference where it was all about how do we get more people interested in this career. So very, very relevant. So thank you. Okay, I'm gonna pull up next. We've got Hamoon.
Okay.
Loading here in one moment. All right, go ahead.
Okay. Can you hear me clearly?
Yep.
Okay. Hello everyone. I'm Hamoon from ISU CBE department and working in Dr. Rule's group. My project title is cell culture and differentiation monitoring using cell scan approach. As you know, cell therapy has been recognized as a novel and effective treatment for a bunch of incurable disease. But the main challenge for development of cell therapy is how to monitor cells during cultivation process and their differentiation. Currently, there are some techniques to monitor cells during their cultivation, like using microscopic-based techniques and also electrochemical sensors, but none of them can work appropriately because, first of all of them are sampling required. It means you need to take sample and then evaluate the sample. So it definitely increases the chance of contamination in your bioprocess, and it's really risky. After that, they just can monitor cell cultivation in a short while, so they are not continuous. And furthermore, they are really expensive. So the idea we are working on is to develop a kind of non-invasive, continuous, and large-scale cell monitoring approach that is highly applicable in pharmaceutical industry, in cell therapy development and manufacturing of cell-based products. So what we have done till now, it's we could develop our cell scanner setup. It's actually mainly consists of a high resolution scanner, and then we tried different parameters affecting on achieving a good image quality. Then we tried to scan different cell species like K562 cells, HMSC cells, iPSC cells, HeLa cells, and you can see the result, the images here. And then we tried to analyze these images using MATLAB segmentor. And what we will expect to do in the future is to try to design a continuous setup for cell scanning and then try it in large scale. After that, we will try to focus on cell differentiation monitoring using our cell scanner. And finally, hopefully, we want to analyze the data using machine learning and determines cells in terms of their count, shape, and size.
Great, thanks.
Thank you.
Opening to questions.
How many samples per day or per hour can this, I mean, it sounds to me like it being a continuous flow system, it's designed to maybe monitor and track a single reactor. Does it have the ability to monitor multiple samples at one time or any sort of high throughput opportunity?
Actually, what we are thinking about is we can develop a kind of bioreactor working with our scanner. As you can see, our setup, it's a kind of casual scanner, but hopefully we can develop a kind of bioreactor and try to have a continuous cell scanning without any sampling. Yeah.
Okay.
So you mentioned the cell therapy application. I'm wondering about this in area that I would refer to as industrial biotech. So for instance, if you took lysine production in a fermenter from E. coli, could you use this technology for contamination detection, for monitoring the health of the E. coli production strain? Any thought about applications there?
First of all, we are thinking about monitoring human cells. It's the first step for us and the main focus we are thinking about. And after that, maybe we can develop it to other kind of microbiology applications, like monitoring other species like bacteria and others.
Great. Thanks, Hamoon.
Yeah.
Go to the next here. Daniela, you'll be next. Okay, when you're ready, take it away.
All right. Okay. Hi, everyone. I'm Daniela Sanchez. I work in biology's lab here at Iowa State University, and my project is on the optimization and development of polyanhydride-based nanoparticle vaccines. So this project is really important and exciting because the overall goal is to create a new vaccine platform and improve on the current available vaccines that we have. And two ways that we're looking into doing this is universality, which refers to using the correct protein to protect against various strains of the same virus. So the three viruses that we're focusing on in this lab is influenza, SARS-CoV-2, and RSV. And the polyanhydrides that we use release this protein over time, which means that our immune system will get re-exposed to this protein multiple times, and this will lead to long-term immunity. So the findings can be applied to the pharmaceutical industry. We are approaching this as a new vaccine platform, but overall, these polyanhydrides can become a new drug delivery platform. And there is one FDA-approved product that does use polyanhydrides already. And then on the right, it's just recent data. So these are SEM images. One of these particle batches, the one on the left, was made using spray drying, which is the way that we're really going to scale up this process. Spray drying just lets us spray bigger batches. However, we do need to work on the aggregation of these particles since we're really aiming for spherical discrete particles. And then the graph on the bottom shows the protein released over time, which shows that this protein is actually released over a long period of time and doesn't all just burst out as soon as it's in, like in this case, it was PBS, but yeah.
Thanks, Daniela. Mike, I'm sure you're well versed in this.
Yeah, I'm in this area. Just one quick question. What are you doing with the samples that you have to confirm uniform size? Are you trying to do that through the spray dry process, or are you using a mill or some sort of post-processing step to create a uniform distribution?
Well, currently with the particles themselves, no, but I know other people in my lab are looking into milling the proteins beforehand so that they're more evenly dispersed and encapsulated.
Okay, thank you.
This one's all Mike, so I'm good.
Thanks, Kevin. Okay, let's go to the next. Thanks, Daniela.
By the way, Danielle, I'm on your floor, so come see me when you get more data.
Okay, yeah.
And if you need a ball mill or other mills, you can talk to my group too. We got all sorts of weird mills. Okay, let's see. We've got, hopefully you say this right, Piyumi out at University of Iowa.
Hi, my name is Piyumi. I'm so sorry about the camera. It's acting up today.
Okay,
So,
that's fine.
So my name is Piyumi Vijayasiriganathan. I'm from University of Iowa. I'm working in Florence Williams lab, and my project is analysis of cellulose from sawdust, and we extracted that from boron Lewis acid treatment. So the main goal of the project is to fully characterize cellulose from sawdust, which is an inexpensive, versatile, readily available biowaste coming from wood and kind of way to recycle and reuse sawdust without going to waste. So from the treatment, we ended up getting lignin and as the solid residue, and then cellulose and hemicellulose in the aqueous media. So my goal is to extract cellulose from this mixture of hemicellulose, cellulose, and boric acid in the aqueous media. And then once I separate the cellulose, actually we have some collaborations, so I can give the cellulose to build up or develop more materials. And the recent data is on the right side. We have several attempts separating boric acid from this mixture of cellulose, hemicellulose, and boric acid aqueous media. And I have used centrifugation and then some organic compounds like pinnacle, catechol to chelate this boric acid to organic layer. And so far it did not work, but I'm investigating more ways to separate boric acid from the aqueous media.
Great, thanks for you. Questions?
Yeah, I'll jump in here. Piyumi, just know that there's work on lignocellulose that goes on at Iowa State as well. Dr. Jinglan Bai has she's done a fair amount of work with nanotubes and onion skins. I'm going to butcher this a little bit. The biosciences has provided some small grants to the BioLab.
Okay.
Yeah, so know that there's this connection. Obviously, lignocellulose as a feedstock is a bit of a holy grail in the whole bio-based space. If I have a question, what about the lignin? This project is pretty focused on the cellulose, but in order to be successful, typically we like to ensure proper value from all of the components. Have you given any thought to the lignin part of this process?
Yes, of course. Another student is working solely on lignin. So yes, I got this project this semester, so I'm working on separating boric acid from cellulose. So I just started. Yeah.
Okay, great, great. Yeah, welcome to the team. Sure.
Great, thanks again. Let's go to our next. Okay, and we got Vamsi, also at University of Iowa.
Hello, everyone. My name's Jet Jaden. I'll be talking about this instead of VAMC today. And so, yeah, can you guys hear me?
Yep, you're good.
Okay, perfect. So our project is molecular separations, and we're trying to do so in a eco-friendly way. So traditional separations like distillation use high temperatures, and lots of energy, and they're also pretty wasteful. And so our goal is to try to find a way to do so without wasting energy and using high temperatures. And we are proposing COFs, or covalent organic frameworks. And so basically how these separations work is that they make a uniform pore size. And you can see in status that TPPA COF has 1.8 nanometer pore size. And we allow molecules to permeate through. And it's basically driven by diffusion, and that the more double bonds in your compound, that the slower it goes through. So methyl stearate has no double bonds, goes through very quickly. The omega-3, which is methyllinolenate, has 3 double bonds and it goes through slower. But this application can be applied for separating these fatty acid methyl esters, which are derived from vegetable oils. And so our recent findings is that we've been using a reverse osmosis separation. And the problem with this is that it's a lot slower. It takes about 5 days. And we're trying to move towards dead end filtration, which is using pressure to push our compounds through the membrane quicker. And this can be done in about 10 minutes.
What scale are you working at currently?
Or like the size of like how many, I guess,
how many-
grams of
Yeah, how
fatty
many
acid.
mils, liters, gallons?
So right now we're working on mill size. Huh?
Yeah.
Yeah, gram scale.
What exact feedstock are you using here?
So right now we just have like 70% pure to 90% pure of these fatty acid methyl esters, and we just we buy them by the vial individually, and we mix them to have an equal molar concentration in our retentate side, I guess, to see the results.
Okay, so it's a synthetic solution, if you will. It's a recipe makeup from pure materials.
I guess. I mean, we have these chemicals separately bought, and then we just put them into our feedstock together, but they don't really interact inside.
Yeah, I'll just throw out the potential value for this technology is in distillers corn oil. So all of the dry mill ethanol facilities all around the Midwest, 40 plus of them in the state of Iowa, have DCO or distillers corn oil as a valuable co-product. It's really a mixture of free fatty acids, the triglycerides, and other valuable components. And if there could be inexpensive ways of separating out this crude oil mixture into the individual components, there would be a lot more value in those individual components compared to the feed. Yep. And if you ever get to the point of needing a distiller's corn oil feed, yeah, I can be a resource. Mike and I can be resources.
Okay, I'll be sure to reach
Yep.
out to you guys then.
Yep.
Great. Thanks, Chet. Thanks for filling in there.
Yeah, no problem.
All right, let's go to the next. All right, Denver is up next.
Can you guys hear me?
Yep, you're good.
Okay. Hi, I'm Denver Landers. I am a first-year graduate student. My PI is Dr. Jarboe, and I'm at Iowa State University. I actually have 2 projects with the EPSCoR project, but I'll only be talking about this one today. We are building microbial factories for the production of trifunctional fatty acids. And our motivation is primarily to reduce our reliance on petrochemical polymers by developing these multifunctional monomers that can be tuned, and through that tuning to improve the feasibility of biopolymers in additive manufacturing. And by expanding this, we should be able to increase the workforce participation. So this is primarily to be used in additive manufacturing of plastics, but I would see it's affect much of the industry as they're monomers. So it wouldn't be like at the fabrication stage, it would be at the chemical material stage. So what we are doing is we will be taking these fatty acids and in engineering these pathways to introduce hydroxyl groups at about the omega-1 position, like at the very almost end of the chain. And we're looking at these maize desaturases that are known to introduce double bonds more mid-chain. And with that, we'll have those 3 functionalities. carboxylic acid, the hydroxyl group for forming the polymer chains, and then the double bonds would be used for tuning the properties by addition of like amines or hydroxylating further, or just an additional functionality for tuning. Right now, we have decided that we will develop octanoic acid, butadecanoic acid, and stearic acid effectively. Those will be our primary targets because we have previous work with octanoic, and C18, stearic is pretty common. And we have selected our strain that we will be developing.
Great. Questions?
Two questions. One, it sounds like this is a bioconversion rather than a fermentation to the target. That is, are you starting with a fatty acid and converting it to the functional version? Or are you, for instance, starting with dextrose in order to make the functional fatty acid?
Yeah, so we would be working with dextrose as the feedstock, and the microbe would do all of the heavy lifting by elongating the fatty acids and the post-elongation modifications.
Okay. And then secondly, once you make the monomer, what's the plan for testing it?
Oh yeah, so we'll do the fermentation, grow up the cells, and then we will do a methyl transesterification to do a gas chromatogram and to analyze where our double bond is inserted, because different desaturases is introduced at different positions, and whether our hydroxyl groups have been incorporated. Is that your question?
I'm wondering about converting the monomer into some sort of a polymer to test its actual polymer functionality or demonstrate.
I see.
Yep.
We will collaborate, I believe, with Jay Wackerly's group in Central College, and they'll do much of the polymer work. Maybe also Dr. Cochran's group here at Iowa State. I'm not entirely certain of that.
Yep, that's the plan. So we'll go back and forth, and yeah, once it's made into polymer, I think Eric's on the hook for doing the mechanical testing.
Okay.
Great. Thanks, Denver.
Thank you.
Go to Brett's next. Okay, Brett. take it
Okay,
away.
can everyone hear me all right?
Yep, you're good.
Okay. So hello everyone, my name is Brett Lange, and I'm a first-year graduate student in Professor Rule's group here at Iowa State University. So I'll be talking about my research project, which is titled Methods for Rapid Expression and Characterization of Nanobodies. So just to provide a little bit of background, nanobodies are a type of antibody fragment derived from heavy chain only antibodies, which are produced in members of the camelid family, such as camels. Similar to antibodies, nanobodies have very high levels of affinity and specificity towards various antigens. And they also have a small size and high levels of stability. And because of all this, they're widely used in different research applications, including in various areas of therapy and diagnostics. So currently, the development and utilization of nanobodies is hindered by slow individual expression and characterization. So this is where my research project is addressed in answering that question. So the first part about that is the expression of nanobodies. So traditional cell-based methods of protein expression are oftentimes very time-consuming and labor-intensive. So one way we can overcome this is by using cell-free protein expression, which is a much more a pretty established method for more high throughput protein expression for screening applications. So far, in my work, I have expressed a couple test case nanobodies in E. coli cell lysate. And then I analyzed those expressions using SDS Page. So you can see in my work, I have the gel output for those expressions with the relevant nanobody band highlighted in red. And so with that, the second part of my project is developing more high throughput process for characterizing nanobodies. One characteristic of nanobodies we're specifically interested in is their affinity towards other antigens. So in order to quantify affinity, one way to do that is by measuring its kinetic parameters associated with the actual binding event of a nanobody to its antigen. So one way I'll be exploring this is through biolayer interferometry, or BLI. So the idea is hopefully I can develop a process where we can directly measure kinetic parameters and binding affinity directly from cell lysate, for example. And then in the future, hopefully I can extend these results even further in collaboration with some other members of the rule group to extend this kinetic modeling using carbon nanotube biosensors instead. So altogether, using cell-free and BLI or carbon nanotube biosensors, the idea is to have a high throughput process for expression and characterization of nanobodies, which can be used to improve upon properties of existing nanobodies or help in the development of novel nanobodies in the future. So with that, thank you for your time.
Mike, Kevin?
Yeah, Brett, nice work. Your example here is the anti-GFP. Are you or any of the others in your lab working with nanobodies that are against infectious agents at this point? And are you able to, if so, are you able to measure in vitro neutralizing activity beyond just the recognition piece?
No, I don't think anyone else in our lab is working with nanobodies specifically. Yeah.
Okay, thanks.
I'm good.
Okay, super. Thanks, Brett. All right, Sarah.
Hello. So my name is Sarah Letourneau. I am currently working for David Verhoeven, and I am in the immunobiology program here at Iowa State. So my focus is more on the immune response and all of this. And specifically, I am focused on influenza. And what I'm going to talk about here is trying to use basically a baculovirus system to produce proteins from influenza that can then go into a nanovaccine polyanhydride system, like what Daniela talked about, to test the efficacy of that vaccine. So why this is important, or like what the need for it is, influenza is a common respiratory virus that circulates yearly, and the current vaccines we use are not, like the efficacy of them is not very good because we are kind of guessing at what is circulating mainly from like the Southern Hemisphere every year in order to develop the yearly vaccines. And that is, for like a lack of a better term, kind of hit or miss every year. So one of the kind of conversations going on within like the influenza A virus world is, how do we develop a universal vaccine? And one of the things that I'm working on kind of in that area is specifically looking at more conserved proteins within influenza. So specifically, I have the nuclear protein, the matrix 2 protein, which is a hydrogen pump on the virus. And I also kind of inherited a neuraminidase aspect of this project. So those are the 3 proteins I'm working with in trying to make constructs within a baculovirus to then produce those three specific they're all separate. They are not all together. They are like three separate constructs, but using all those three, trying to produce those proteins, like isolate them, purify them, and then send them over to be developed, like put into the polyhydride nanovaccine work that Daniela was talking about. The goal there is to produce, as she said, like a slow release vaccine and see if that gives us an overall better immune response when put into animal models, to then potentially go down further in the line. Hopefully one day, if those all work well, would be to go into with humans. So basically currently where I'm at is I have all of those three in baculovirus, currently trying to get them to express in insect cells, like isolate the protein and get it into a vaccine. So that's where I'm sitting at right now, working on that expression. And then the next steps quickly are isolate and purify those proteins, get them into a vaccine, and then test efficacy in animal models and compare the effectiveness of that polyhydride system versus the vaccines we commonly use today. That's what I got.
Great, thanks, Sarah.
Yeah, sir, this Mike, it's your good news that you're not using the HA on this. That's tricky with the monomers, dimers, trimers and stuff.
Yeah, well, that's with the matrix. That's one of the issues because it's like a quadrivalent protein, but yeah.
Yeah, so do you know, not only the sequential part of this, but do these antigens, are they like HA, that conformational structure is important in their immunogenicity? And if so, what are you going to compare baculo to CHO or other systems to see that have different glycosylation patterns to see which one is optimal?
So I know it's the nucleoprotein, that is not necessarily an issue. The matrix, that M2, as I said, because it is a quadrivalent protein, that is an issue we've already ran into. And so we have kind of gotten a like specific sequence to start that with to hopefully be able to produce it as a quadrivalent protein. And then I know the neuraminidase is a trivalent protein, so that is one we're keeping our eyes on. I don't believe we plan to compare it to other systems, but I know we want to compare it to other vaccines that we would use.
Okay. Just a heads up, I don't know if it'll end up with this, but in a previous life, I developed a bivalent vaccine, those H1N1 and H3N2, and the H1N1 was extremely stable, even in the trimeric form. And in the same system, which happened to be baculovirus, the H3N2 wasn't. So there is some isolate to isolate variation within flu on these antigens.
Yeah. Yeah. Keep it in mind. Flu is tricky.
Great, thank you, Sarah.
Thank you.
Okay, let's go to all right, Mohammed Noor.
Good. Hello, can you hear me?
Yep, you're good.
Okay. Hi, I'm Noor, a first-year grad student from Clothier Lab at Iowa State University. So today I'll be talking about a computational study to predict the future variants of SARS-CoV-2 that can escape our body's immune response. So the majority of work has started before I joined. So I'll be going over what has been done till now. And I think Ratul has spoken with about this with Mike, and also he's in touch with E from OIPTT as well. So basically, there's the virus binds with human receptor protein called ACE2, and it basically starts infection from there. And our immune system basically makes antibodies that binds with those viruses to neutralize it. So virus has to come up with mutations that serves two purposes. It has to decrease the interactions with the antibodies, as well as increase the interactions, or at least maintain interactions with the ACE2 protein itself. So it's like playing two chess game at the same time, using the same white pieces, using the same moves, but having two different objectives. For example, win in one game or lose in another one. So the SARS-CoV-2 virus is doing this with ease, and we're trying to capture that. And predicting potential mutations that might appear in future variants will help us to come up with antibodies, antibody and neutralizing antibodies before the virus reach potential frequency, or let's say mortality rate in global populations. And at the very onset of the pandemic, the virus was gaining 2 mutations per month. So a region of the virus that actually mediates the entry of the virus in the human body's receptor binding domain. This is highly variable region. So, and we evaluate the future moves of the virus as it mutates itself and try to predict future variants that will escape the immune response that we administered today, or increase or maintain the human response, interaction with human response protein. And to do that, we started off with a commercial antibody, a LICOV 1404 bound to the spike protein. That was our starting point, and we used a combination of integral programming, heuristic search, and molecular biochemical scoring of binding affinity between the evolving spike, the antibodies, and the entry receptors. And that led us to, for initial simulation, that led us to about 86%, to predict about 86% of the circulating variants. So this model was not fed any info on what the potential infected virus could look like. So it was kind of a blind prediction. And as you can see in the plot that I've added in the slide in the right-hand side, so basically we're plotting the interaction energy, binding interaction energy of the viral protein with antibody on the x-axis and the human receptor protein on the y-axis. And the lower the value, the stronger the interaction is. So all the blue dots are from our simulations, and we kind of also added the mutations that actually appeared in real life. Those are in oranges. As you can see, a model can come up with not only mutations that appear in real life, but also some more that might appear. And most of the mutations are clustless, so that's why it might look like that. There are only few oranges. Basically my work will be heavily focused on modifying the integer model, maybe changing the integer model into a more sophisticated one, let's say a Monte Carlo search method, to kind of make a better version of this. Yeah, that's it.
Great, thank you.
Yeah, so this is kind of a new area for me. I'm very interested in it, and as you said, I've met with Ratul. I've also learned that depending on the algorithm you use, the results can be different. And so are you taking multiple algorithms and requiring that 2 of the 3 show an outcome before you chase it, or are you using a single dedicated algorithm?
Right now we're dedicating only one algorithm.
Okay.
Yeah, and we'll be this is our initial findings that we did. All the simulations take some time to finish because it's an iterative process. So, and then after we feel like we have like a good data set and that to build on, that the entire the pipeline actually works, and then we'll move on to like a more advanced or more sophisticated algorithm, like for example Monte Carlo search method that I already mentioned.
Great, thank you, Mohammed. Okay, go next. All right, next up is Binayak Pandey.
So hello and good evening, everyone. I'm Binayak Raj Pandey. I'm PhD in microbiology student at Dr. Chunui Xiang's lab at Iowa State University. So my project basically revolves around biofilm formation, and nanocellulose production, basically a nanomaterial. So what is the motivation behind my research is we have a nanocellulose, and we know that this has been extensively studied, and there are data that suggest that it could be better than Kevlar when in the strength or elasticity. But nanocellulose can be produced by top-down approach. That's what we know. We take plant cell and then we make fiber out of that. So that is top-down approach. So here we are adopting the bottom-up approach where we are using bacteria to produce nanocellulose. So we have better control over these processes. So why nanocellulose, especially bacterial nanocellulose, is because it's strong, it's flexible, it's biodegradable. That means it does not have any effect in the environment, and it's biocompatible. That means it does not cause any kind of allergical reaction in the human body. So that leads us to application part, where after developing the bacterial nanocellulose production, we are targeting it to test in healthcare sector, where we will see how it's effective in drug delivery, in tissue engineering, and obviously we will produce advanced composites. And there are some collaborators, I think, who are interested in electronics and biosensors. Our lab will also be dealing with sustainable textile materials, so how to produce that. So we have this technical electrospinning. So we can use electrospinning to produce a defined fiber out of that. And then we can use this nanocellulose to produce inks that are used in 3D printing in packaging material. So this single material has many applications, and talking about medical point, it's pretty much applicable because it's biocompatible. So current status is we are using Comagati gylinus to produce this nanocellulose, which has been pretty extensively studied as a model organism to study plant cell wall. So what we are doing now is we are trying to produce a new and optimized media so that we can get maximum yield to produce this nanocellulose. So in image, you can see we have before harvest image. So it's pretty thick. So it generally floats over the liquid surface. So after harvesting, we treat it and then we dry it out. After that, it becomes very thin. And we are trying trying to know is like which carbon sources will give better result for now. And then we'll try with different salt and protein combination to get better results out of it. So currently, like we are done experimenting with carbon sources like fructose, sucrose, glucose, even like mannitol, and the glycerol. So glycerol don't give that much of optimized result. So I think we'll be more interested in fructose, sucrose, and glucose. And another thing is we are doing a laboratory evolution where we are training bacteria to utilize mannitol. So in the first instance, this bacteria could not utilize mannitol to produce thicker biofilm. So now after training, so after laboratory evolution, these bacteria can now produce thicker biofilm in the form of nanocellulose. And another interesting thing we found is this bacteria can also grow in ethanol as a sole carbon source. That means it will have a future advantage where we can take waste as a biomass, then convert it into ethanol, and then feed the ethanol to this bacteria to produce nanocellulose. So we are still studying its behavior, and then analyze the media, which media will go this year. After that, like we have other pipelines will follow. Thank you.
Thanks, Binyak. A couple questions again on the production. I think I heard you say biofilm. So
Yeah.
is this a thick biofilm?
Yeah, thick biofilm, but nanocellulose will be there.
Yeah, and so then is that a mixture of the actual nanocellulose with the bacteria? It's a mix?
We have seen it under microscope. So here, certainly like some bacteria get attached, but the treatment process, the downstream process we are following will make sure this bacteria will be dead. So like we are boiling it, we are treating with sodium hydroxide, and after baking soda and all. So definitely we'll get rid of these bacteria, but bacteria will be present, of course, because it grows in very close contact with the bacteria. So there will be bacteria all over the liquid. So whenever we take these out, this kind of unavoidable situation now. But yeah, so but one of the good thing is this Comagotibacter gyrus is bacteria that can be consumed. So I think you have heard about kombucha that we drink, the probiotic. So this is not harmful to human if consumed. So I think that makes this experiment a bit different because like this can this nanocellulose don't come from any kind of sort of pathogenic bacteria or any bacteria that has the pathogenic advantage over the human host.
Yeah. I'm a little unclear from the graph on the lower right, whether a higher number means a better substrate or a lower number is a better substrate.
So this graph has been designed after we have dried the sample. So in the image you can see it's just after harvesting. So that means it has not been dried. So after we dried, we have taken the dry weight. And since we used like 20 gram in 1 liter, that means now we are getting around 2 grams. So that means conversion rate is 10%. So we are getting 10% of nanocellulose from whatever substrate we are trying. So I think, yeah, this graph represents that. And since like you can see standard deviation as an error bar there. So that means there are some factors which governs how these nanocellulose or how thick this nanocellulose will be formed. So, but for now, I think we are first analyzing the effect of carbon source, and then like we will figure out how other factors will influence like pH and temperature.
Okay,
great. Thanks.
Yeah, sorry, Nigel, just
Yeah.
one more point. I just throw out for Iowa, glucose from corn is going to be your most available, most consistent, lowest cost feedstock.
Okay.
Super. All right. Thanks, Binyak. All right. We've got 2 left. Richardson. So
that would be me. I don't have a camera on this computer, unfortunately.
Okay, Sam, you're up. Oh, okay, go for it.
Cool. Well, you will notice some similarities between me and Denver's projects. We're both part of the EPSCoR grant here at Iowa State, but I'm working under Dr. Shao. So my project focuses on building microbial factories for the synthesis of trifunctional organic acids, specifically edeconic acid. So traditional microbial factories using yeast are easy to maintain at industrial levels, but they have production rates that are a lot lower than are desired in these industrial settings. So to solve this issue, we need to either shorten the course of production significantly or increase the titer dramatically. So we had 3 organisms that were attractive for this work, being Ustalagomatus, which is the corn smut fungus, Aspergillus terreus, which is a soil fungus, and Vibrio nitrogens, which is the fastest growing bacteria to date. Ustalagomatus and Aspergillus terreus have high titer, but they are both filamentous fungus, so there may be some issues in scale up. So the reason why we want to increase our edeconic acid production is due to its role as a precursor in the synthesis of resins, fibers, adhesives, thickeners, and binders, along with its superabsorbent properties in copolymers, which is highly valued in sanitary items and things like diapers. So there's a $260 million market value projected for 2025. So this research has potential to increase production and profitability. For some of the work involving edeconic acid as a copolymer, we're sending this over to Cochrane's group. So the research is in its early stages, but we performed a risk assessment for our organisms of interest. V. nitrogens is fast growing, so it's an ideal cloning strand. However, its ability to both grow well and produce edeconic acid is suspect, as the bacteria does not function well under low pH, which would be a consequence of our product being an organic acid. So Oustulagomatus and Aspergillus terreus present better options for production, though the scale-up will be a challenge due to the highly branched filamentous nature of each species. So between these two, we chose Oustulagomatus as it offers a unique opportunity to develop new genetic tools, since there is a lot fewer tools available for it than Aspergillus terreus, while still advancing the production of edeconic acid. So currently I'm exploring available genetic tools and genetic libraries for relevant plasmids to perform transformation on Oustulagomatus.
Very great. Kevin, Mike?
Yeah, so you mentioned pH, and I would second the fact that some organic acids that are made need to be pH controlled, and effectively you're making the salt rather than the free acid, which is lower. The salt's always lower value, and it really adds to the DSP in order to convert to the free acid. The Oustulago, does it tolerate low pHs?
It does not, but it does a lot better than the other two. And we may be able to pH control it a lot easier than our other two species.
Okay. And then secondly, it's just that there have been attempts to commercialize edeconic, and I assume that you've done some mining of the literature to understand what other strains have been used, and maybe most particularly what the benchmarks are for yield, titer, and productivity.
Yes. So off the top of my head, the numbers that I can remember are that Aspergillus terreus is the general species that we use. So Aspergillus terreus is the general species that we'd use in the lab. Yeast is our main producer industrially because we know how to manipulate its conditions the best. Let's see. So essentially, we can manipulate its conditions the best, but it does not give us a large titer. Aspergillus terreus gives us about 150 grams per liter, while Oustulagomatus gives us 200 grams per liter. So we have a better titer from Matus than terreus, but the conditions are more difficult and there are less available tools, which is why Dr. Shao decided to take this on, because her group is focused on using non-model organisms.
Great. And we have one more presentation left. I think it's Chris who got queued up here. All right, Chris.
All right. So we are kind of going to switch gears a little bit. We have a very fascinating project indeed, in the fact that instead of using bacteria to produce a specific compound of interest, or like a product, for instance, we are actually trying to create bacteria as the end product itself. So essentially what we're doing are creating what are called bacterial ghosts. And bacterial ghosts are empty bacteria that are completely devoid of its internals, but whose surface components remain intact. So the important thing about this is these surface components can interact with different receptors found throughout the body. And these interactions can lead to increased adhesiveness. So you can actually target specific, or bacteria can target specific receptors which allow them to localize in a specific area. Now, bacteria ghosts have been used in a variety of different applications, including in terms of drug delivery. But the thing that we don't really know is the extent that they interact with receptors. And there's one particular class of receptors we're really interested in. They're called G protein coupled receptors, or GPCRs. So GPCRs are a superfamily of proteins. They have a very accessible active site in the extracellular space of the human body. And they're found throughout the entire body, whether it's the lungs, the kidneys, the central nervous system, they're found everywhere. And GPCRs also are often overexpressed in certain disease conditions. So if you combine some of this information, they're very, very attractive targets for drug delivery. So the point of one of the big points of this project is to find out or try and probe the ability for bacterial ghosts to interact with these GPCRs. And eventually we want to use this as a tool for drug delivery, ideally with enhanced targeting ability, allowing us to actually deliver drugs directly to a disease site rather than have it spread throughout the entire body. So there's been quite a bit of research going into this so far. So the first thing that we wanted to do was essentially build a platform. We wanted to use just E. coli, since there's a lot of data about it. Everyone loves to work with E. coli. So we've used 3 different E. coli strains, 2 different commensal strains, so bacteria that have been isolated from the human body, and then 1 probiotic strain, so a E. coli strain that's actually been shown to be beneficial to human health. So with all 3 of these strains, we were able to chemically produce these bacterial ghosts. We confirmed this by, first of all, looking at DNA release. This was used as a proxy to determine if the internal components were being released. And during the chemical treatment, the DNA was completely released, whereas in our final ghost, after extracting any DNA that could potentially be remaining in the bacterial ghosts, there was no DNA whatsoever. So we're just getting some confirmation that these internals are released. And if there's no genetic information, these bacteria can't grow or reproduce or anything like that. So even though the internal components are released, we wanted to see if the membrane was still intact. So we use SEM just to visualize the overall morphology of the bacteria, the chemically treated bacteria. And we did notice that the membrane and the overall morphology was still intact, which is again suggesting that the membrane itself was still there. So again, we extended this to 3 different E. coli strains. So this is just one particular strain, but we've shown this works for 2 others as well. Now, the final piece of information we really wanted to start looking at is using high throughput screening techniques. So as I've mentioned earlier, the extent that bacteria can actually interact with GPCRs is relatively unknown because there's hundreds of potential GPCRs you can investigate. Conventional techniques is just unfeasible, both in terms of money and also in terms of time. So if we're able to adapt high throughput screening methods, we're able to look at a lot of potential GPCRs simultaneously. As far as we're aware, there's been no other study that has applied high throughput approaches for whole bacteria. So this is very much the novel part of this project. And essentially we looked at 3 different concentrations of bacterial ghosts and noticed that there was one particular concentration. It was interesting because it was the middle concentration. So there's almost a Goldilocks zone in terms of bacterial ghost concentrations required to bacterial ghost concentrations to be applied to in a high throughput method. So with this high throughput method, we found 3 potential interactions. And of course, there's a lot of work that still needs to be done to actually prove that these interactions are truly interactions and not just one-offs from this screening. So eventually this was initially done with one bacterial ghost strain, and we plan on doing this with the other 2 E. coli strains that we've shown to create bacterial ghosts.
Great. Opening it to Kevin, Mike.
Yeah, just throw out, this is super cool. I've learned a lot here this afternoon. Never came across the term bacterial ghost. So thank you, Chris.
Of course, it's again, it's a very interesting project that the bacteria itself is the product.
Yeah, Chris, during the chemical process, without having a viable organism, now your membrane permeability changes. How are you able to load your bacterial with your target of interest? And is there a way to increase the efficiency of that target? And then also, what's the stability of these ghosts over time? In a therapeutic thing, these might sit on shelves for weeks or months. How do these ghosts decay over time, or how do you manage the stability of product within the ghost itself?
Sure. So during the chemical treatment process, theoretically, you're creating pores or little holes in the membrane itself. So in terms of the drug loading, what most people have seen is that the drug naturally diffuses into the bacteria itself. We are planning on looking at drug loading, and we haven't gotten around to it quite yet. But as far as I'm aware, the biggest things are going to be due to things like the buffer that you're using or the solvent that you're using during the drug loading process. Again, we haven't dug too deep into the drug loading itself quite yet, but outside of just letting the drugs naturally diffuse into the bacterial ghost, I don't know if there's a way to artificially pump those numbers up, essentially. Now, in terms of the stability, I haven't seen much studies that have actually looked at the stability. What we do at the very end is we freeze dry these bacterial ghosts. So I know we've been able to use these after they've been freeze dried, months after the drying process, so that they aren't going to degrade like rapidly where you have to use it within weeks. But we don't have definitive data that be like it lasts 6 months before you use it.
All right, thanks.
Great. Thanks, Chris. And since that's the end, I'm going to stop sharing here and thank everyone for their participation. That was super helpful. I mean, just also from my standpoint of seeing what everyone's working on, and I already have written down interaction points that I want to follow up on. So thank you for that. I'm going to ask Kevin and Mike to hang on just for a second, but excuse all the students, and thank you for your hard work on this. Appreciate it.
Yeah, thanks everyone.
Thank you.
Thank you for your time.